Okay, so we're moving on to Chapter 3, Mendelian Genetics. I think some of this will be a review for you from general biology, but we're going to delve a lot deeper into Mendelian genetics than probably what you covered in biology. And this is actually a pretty in-depth chapter. I'm breaking it up, I think, into 4 different lectures, but make sure, really take your time going through these lectures and read the chapter in the book, work some of the problems at the end of the chapter to get a good understanding of this, because we're going to be building on this in chapter 4. So if you don't get the material in chapter 3, then it's going to be that much more difficult to understand the material in chapter 4. Okay, so just an introduction. We have Gregor Mendel, and we've already talked about him and about his genetic studies with pea plants. Through his research, he was able to determine that there were these little discrete units of inheritance that were passed on from parents to offspring. And his work formed the basis of what we now call transmission genetics, which is how we study how genes are transmitted from parents to offspring. So in his experiment, like I said, he used pea plants, and it was a really good model system because the plants are easy to grow and they will grow to maturity in 1 season, so it doesn't take a lot of time. You can have true breeding strains, and you can control crosses of the different traits through self-fertilization or cross-fertilization. So you can allow the plants to self-fertilize, and that will keep them breeding true. Or if you want to cross-fertilize between 2 different plants, like say for example a tall plant and a short plant, then you can do that and you can start mixing up the genes. And most importantly, it has observable characteristics with 2 distinct forms. I think Mendel would have gotten really confused if there were multiple forms, more than just 2 different alternatives. So this is what he studied. You can see the different characters. There were 7 different characters. So we have seed shape, seed color, flower color, stem height, all of those. And then for each character, there were exactly 2 contrasting traits. So like I said, if there had been multiple, like the peas could be green, yellow, or brown or something, that I think that would have made it a little more confusing for Mendel. But luckily that was not the case. So when we talk about true breeding strains, what really do we mean by that term? A true breeding strain is going to only display that trait that you expect it to over and over and over again through self-fertilization. So if you have a tall plant and you allow it to self-fertilize itself over and over and over multiple generations, it's always going to produce tall offspring. It's never going to throw a short plant in there. When they say they breed true, it means that they always have the same phenotype over and over with each generation. And really what that means, and you'll see later down the road, is it means that it's homozygous for that particular trait. So the difference between self-fertilization versus cross-fertilization, you can see here in the diagram that there's 2 main parts to a flower. You have the girl parts, which is the pistil here, and then you have the stamen, which is here, or these are the anthers of the stamen anyway. And so this is the girl and this is the boy, and the boy parts has the pollen, and so the pollen ends up in the girl parts, and then and that's how you get a new plant. So with self-fertilization, you just allow that to happen. If you don't want the plant to self-fertilize, then you remove those anthers so that there's no pollen there to fertilize the plant, and then you can take anthers from a different plant and fertilize the pistil on purpose, and that's how you do cross-fertilization. So Mendel studied 1 character at a time, kept very meticulous records. I guess as a monk he didn't really have a lot to do, so he had plenty of time to play with his pea plants. He spent years on this research, and published it, and nobody really cared at the time. But at the turn of the century, in the early 1900s, his work was rediscovered. Some other researchers ended up doing the same kind of research and coming to the same conclusions. And a big part of doing research is you go and you look at literature, published literature, to see what other researchers have published. And so these researchers recognized that Mendel had already discovered all this. And so he finally got the credit he deserved for all the work that he did. And his postulates were eventually accepted as the basis for transmission genetics. And in fact, he was such a big part of that that we also call it Mendelian genetics. So let's first talk about the first type of experiment that Mendel did, which was a monohybrid cross. So a monohybrid cross is going to study one trait and see how it gets transmitted from generation to generation. So we've only got one pair of contrasting traits for one character. So for example, if we're looking at flower color, one plant would have white flowers and one plant would have purple flowers. And so we're going to cross them and see what kind of progeny we get. The first strains that you use, so like the white flower strain and the purple flower strain, they're going to be true breeding, and we will cross them, and that generation is considered the P1 generation, or the parental generation. The offspring of that initial cross is called the first filial generation, or the F1 generation. And then in the F1 generation, we just allow those plants to self-fertilize. It really would be the same thing as if we crossed between two plants within the F1 generation, because they all should be uniform, but it's much easier just to let them first self-fertilize versus removing the anthers and all that business. So we just allow them to self-fertilize, and that their progeny are the F2, or second filial generation. So Mendel, one at a time, took all 7 characters and performed parental crosses and F1 crosses and analyzed the results. So in the first cross, in the first monohybrid cross with the parentals, he noticed that when you cross a tall plant with a short plant, all the progeny end up being a tall. So the first question he's asking is, what happened to the short plant? You got to think about the different things that maybe he didn't know what was going to happen. You know, he didn't know anything about genes and segregation and all that kind of stuff. So what would you think if you took a tall person and a short person? Would you think their children would be all tall? Would you think they would be all short? Would you think, you know, probably the most logical thing, if you didn't know anything about genetics, is you would think they would be something in between. Like they wouldn't be as tall as the tall plants, but not as short as the original short plant, something in the middle. And that's not what he got at all. He got all tall plants. So that was a little bit confusing to him at first. So what happened to those short plants? What happened to that short trait? So then he allowed those plants to self-fertilize and to produce the F2 generation. And he noticed in the F2 generation that that dwarf plant showed back up. But it was always in a much smaller number than the tall plants, and he actually, through counting them up, he recognized that there was a very consistent ratio of 3 to 1 of tall to dwarf. So three out of every 4 plants in the F2 generation were tall, and 1 out of every 4 plant, or 1/4, or 25%, you know, just depending on how you want to describe it, were dwarf plants. And he did the same thing with all of the characters, not just the height of the plants, but the seed color, the seed shape, the flowers, you know, everything. And he got the same results every time. He also switched it out. He didn't know if it had anything to do with female or male, if that made any difference. So, for example, you know, maybe the first time he had a tall male and a short female, and then, you know, looked at those results. And then the second time he would do a tall female and a short male. And he found that's called a reciprocal cross when you switch the sexes. And he found that it didn't make any difference at all to the results. Now, we'll see later on in the semester that it does, for some traits, it does make a difference if it's male or female, but for the traits that Mendel studied, that wasn't the case, which is probably a good thing, because that would have just made it confusing for him. So he proposed the existence of these unit factors, which we call them genes, but at the time he didn't know anything about DNA or chromosomes or anything like that. So he used the term unit factors to describe some physical entity that's getting passed down from parent to offspring, and that's serving as the basis for heredity. So with everything he learned from his 7 different characters doing monohybrid crosses, he came up with 3 postulates. The first postulate states that that these unit factors exist and that they exist in pairs. So he was able to figure out that all of these plants had what we call diploid cells, which he probably didn't use the term diploid, but we know that diploid means you have 2 copies of every gene. Well, he figured out that these plants had 2 copies of every unit factors for each character that he was studying. And so if you have 2, and they're 2 different ones, and you have 3 possible combinations. He also recognized that there was with these traits, with these factors, that one was always going to be dominant to the other. And so if they were both present at the same time, like they are in the F1 generation, that one will always be dominant to the other one. So the tall factor is going to be dominant over the stem factor, and so all the progeny are going to look like one of the parents, and that would be the one that was carrying the dominant factors. And then he was also able to figure out that during the formation of gametes, so during the formation of the pollen and the pistil, that these unit factors actually separate or segregate randomly, so that each time a new plant is formed, it's completely random, and they have equal chance of picking up one trait or the other, or one factor or the other, with equal likelihood. So let's go over what we know, the terms that we use to describe the data that Mendel obtained. So when we talk about the physical expression of a trait, like purple flowers or tall plants or round seeds, we're talking about the phenotype. We call that the phenotype. And these unit factors that Mendel discovered, we call these genes. We know that these are genes that are present on chromosomes. The phenotype for any given character, so flower color, height, seed shape, these are all determined by alternative forms of a gene, which we call alleles. And we know that each plant will have 2 forms, 2 alleles. So every diploid organism will have 2 alleles for each trait. One that obtains from its mother and one that obtains from its father. So if a plant inherits 2 of the same alleles, then that plant is considered homozygous for that trait. If it gets 2 different alleles, then it's considered heterozygous for that trait. And if the organism is heterozygous, then the form that trait, the phenotype of that trait, is based on which allele is dominant. So for example, in the tall and the dwarf plants, the tall allele is dominant over the dwarf allele, so a heterozygous plant will appear tall. All right, just some terminology and symbolism. For Mendelian genetics, you always use the first letter of the recessive trait. Now, so this book says, I know when I was in college, and I think even previous books that I've used to teach this course, they use the letter of the dominant trait. So it probably varies depending on which textbook you're using. We'll go with this textbook so that it doesn't get confusing, but it really doesn't matter which letter you use, honestly. You could use whatever you wanted as a symbol as long as you were consistent. But so we'll just stick with the book though. The dominant, so for the tall versus short or dwarf trait, the dominant allele would be designated with capital letter D. Recessive allele would be a lowercase d. Usually it's italics as well. And So what two letters, so what two alleles each individual plant carries is what makes up its genotype. And so when you write out the letters, you're writing out the genotype. So if you had two alleles for the tall plant, then it would be two capital Ds, and that's homozygous dominant. If they are heterozygous, meaning they have one of each allele, then it's big D, little d. And if they're homozygous recessive, then that means it carries the two recessive alleles, which for the dwarf plant, which is the two lowercase d's. And so the only time you see the recessive phenotype is if it's a true breeding plant and is homozygous recessive for that trait. Okay, this is a little animation that came with the book that kind of goes over everything that we've been talking about so far. So like we've said before, Mendel had true breeding strains for each of the 7 characteristics that he was looking at for each trait. So he had tall plants that were self-fertilized over and over and over again and would always produce tall plants. And same thing with dwarf plants. And as long as they're self-fertilizing, they'll breed true. Then he could cross-fertilize between the 2 different strains to see what those progeny would be. So here you can see, through self-fertilization, a tall plant, all of its progeny are going to be tall. And then in the next generation, all of those are going to be tall. And on until you cross-fertilize. So the first cross-fertilization that he did was the P1 parental cross, where he took a true breeding tall plant and crossed it with a true breeding dwarf plant. And all the progeny that was in the F1, the first filial generation, were all tall plants. So the dwarf trait was absent from the first generation. Then he crossed or allowed the F1 plants to self-fertilize, which is kind of the same thing as if the you cross-fertilize between 2 plants within the same generation, since they're all uniform. So he allowed these plants to self-fertilize, and in the process, he noticed that the dwarf trait reappeared in the F2 or the second filial generation. And it was always, when he did this with all 7 characteristics, it was always the same ratio. It seemed to be about 3 to 1 of one trait over the other. So in this case, it was tall plants to dwarf plants. And he concluded from that there must be some kind of unit factor, something that's getting transferred from parent to offspring, that is the reason that he got these results. So the postulates that he came up with, his first postulate was that there were these unit factors that exist in pairs in individual organisms. And that was, like I've said before, it's just amazing that he came up with that all by himself without any knowledge of chromosomes or genes or even cells, really. And so here we can see the unit factors for the true breeding tall plant would be 2 factors for the tall trait, and a dwarf plant would have 2 factors for the dwarf trait. And then in the F1 generation, we have tall plants that have the 2 traits, the 2 unit factors, but only display 1 trait. So they have tall plants, but they have 2 different alleles. And so he also postulated that there would always be 1 trait or 1 unit factor that was dominant over the other unit factor. And in a case where a plant carried both factors, the dominant one would determine what that phenotype would be. And his third postulate stated that when the plants undergo meiosis, which he didn't know it was meiosis, but when they formed gametes, that all of the different unit factors would segregate independently, and that the new progeny that was formed from fusion of gametes would have a 50-50 shot at picking up either one of those unit factors, which we now know are alleles. Okay, so let me see. Okay, so now we're going to talk a little bit about Punnett squares, and I'm sure you were introduced to Punnett squares in your general biology class, and you probably know Reginald Punnett was the guy that figured out how to create these squares to be able to easily look at these crosses and the results of these crosses. And monohybrid Punnett squares are pretty easy. So what you do is you have to know what the genotypes are for the two plants that you're crossing. So in the parental cross, we know we had a homozygous tall crossed with a homozygous dwarf. So we had a capital D crossed with a lowercase d. And we know that the second thing when it comes to a Punnett square is you have to know what the gametes form are. And so when you've only got one character that you're looking at, that's pretty easy. We know that there's only two possible gametes produced by each parent, two capital D gametes for one parent and two lowercase d gametes for the other. And so you just write them in the Punnett square, and you can see here, this is actually, and this is just by convention, the male is going to be on this side, and then the female is across the top here. So this is the male gametes 1 and 2 and female gametes 1 and 2. Once you've created that with your gametes, then the next step is simply to perform the cross. So you look and you say, you just fill in the grid. So you look and you say, okay, a big D, little d, put it together, that's a heterozygote. And you just do that in all, empty spaces, in every empty space in the grid. And then that's all of your progeny. Those are all the possible genotypes that you could get from a cross. And then it also gives you the percentage or the probability of, what percentage of the progeny would be. So in here, all 4 are going to be heterozygous. So 4 out of 4, or 100%. So the phenotype of the F1 plants is all tall. Now when we do the F1 cross, this time we're looking at 2 heterozygotes. So you can have 2 big D, little d's. They're both tall phenotypically, but they are heterozygous genotypically. Then when they form gametes, you can see each individual plant is each gamete is going to have either a big D or a little d. So it's either going to have allele for a tall plant or a short plant. So when you create the Punnett square this time, you put big D, little d here, and then big D, little d across here. And then you can see when you fill it in, you have 2 big D's, so you have 1 homozygous dominant, you have 2 that are heterozygous but still show the dominant trait, and then you have 1 recessive. So in this case, 1 out of 4 is going to be dwarf, and 3 out of 4 are tall, and so that's your phenotypic ratio is 3 to 1. Your genotypic ratio, though, you have one dominant homozygous, 2 heterozygous, and 1 recessive homozygous. So you get a 1 to 2 to 1 ratio for the monohybrid F1 cross. Okay, so just a little talk about these, what we mean when we say probability. So when he did these crosses, he noticed that every time he got the same results, that you would always get about 75% of the dominant phenotype and 25% of the recessive phenotype in the progeny in the F2 generation. So meaning 3 out of every 4 plants had the dominant trait, and only 1 out of every 4 plants had the recessive trait. So you can predict, so he was able to predict in terms of probabilities what each offspring would have every time he did a cross. Even before he did the cross, he knew what the results would be based on these ratios. And we're going to be doing a lot of those predictions moving forward. So far we know for monohybrid cross, if you take two true breeding plants, one's homozygous dominant and one's homozygous recessive, all of the plants in the F1 generation are going to be dominant, and they're all going to be heterozygotes. And then when you perform the F1 cross, when you cross two heterozygotes, you end up with that 3-to-1 phenotypic ratio and a 1-to-2-to-1 genotypic ratio. So the last type of cross that we're going to talk about in this lecture is the test cross. Now, and we're still doing it with just one character. So we know that when you take a true breeding plant that's dominant and cross it with a true breeding plant that's recessive, you get all dominant. If you take two heterozygotes and cross them, you get that 3-to-1 phenotypic ratio. Well, how can you tell? Let's just say you've got a tall plant and a short plant, and you want to cross them. How do you know what the genotype of that tall plant is? Of course, you know that the short plant is homozygous, because if it's showing the recessive trait, it has to be homozygous. But what about that dominant one? How do you know? It could be heterozygous, or it could be homozygous. You don't know. So in order to determine if the plant is heterozygous or homozygous, you can perform what we call a test cross. So a test cross involves crossing the individual with a dominant trait with a homozygous recessive individual. So if it's homozygous dominant, then you know that all the progeny will have the dominant phenotype, because that's basically just a parental cross like we've already discussed. So we've already done that before, and we know that the F1 progeny are all heterozygous and show the dominant phenotype. But if the genotype of that tall plant is heterozygous, then you're going to get a different ratio. So you'll note here what happens is there's really only two kind of combinations that you can get, and it'll be a 50-50 shot. Half of them will be tall and half of the plants will be dwarf, and so you'll get this 1-to-1 ratio. And so that's how we know. So if you do the cross and you end up with a 1-to-1 ratio of tall to dwarf plants, then you know that that original tall plant that you were looking at was heterozygous. And that's called a test cross. Alright, so just a little review about these monohybrid crosses, because these numbers are important. As we move forward, it's good to almost memorize these different ratios. So just remember that when you do the parental cross, there's really no numbers to memorize. You get all heterozygotes and they're all dominant. But when you do the F1 cross, memorize these ratios: a 1-to-2-to-1 genotypic ratio and a 3-to-1 phenotypic ratio. And then a test cross gives you that 1-to-1, both genotypic and phenotypic ratio. Okay, so that's it for this first lecture. So go ahead and answer the review questions, and then we can move to the second lecture where we'll start to talk about dihybrid crosses.